Thank you, Elisa, for this very kind introduction. And like you said, today I'm going to present unpublished work on how mutant KRAS alters the cellular and transcriptional landscape of lymphatic vessels. Before I present my lab's work, I just want to briefly review the organization of the lymphatic network and the zonation of lymphatic vessels. The lymphatic network begins with lymphatic capillaries, which are comprised of live one positive oak leaf shaped endothelial cells with button-like junctions. And these vessels are the absorbing vessels of the lymphatic system, which pick up fluid, macromolecules, and cells from the interstitium, and then transport this fluid as lymph to the second type of lymphatic called a collecting lymphatic vessel. And collecting lymphatic vessels are comprised of live one downregulated, elongated endothelial cells with continuous, or what we refer to as zipper-like junctions. And collecting lymphatic vessels contain intraluminal valves, which partition the vessels into discrete contractile elements called lymphangions. And the pumping of lymphangions propels lymph to lymph nodes and ultimately to the thoracic duct, which empties into the left subclavian vein. And complex lymphatic anomalies, as everyone here is aware, are rare lymphatic diseases caused by the maldevelopment of these vessels. And we and others have recently reported that complex lymphatic anomalies can be caused by somatic activating mutations in KRAS. And KRAS is a small G protein which functions as a molecular switch. It is inactive when bound to GDP. When bound to GTP, it undergoes a conformational change, which allows it to interact with downstream effector proteins to promote signal transduction. And two pathways commonly activated by KRAS are the PI3 kinase and MAP kinase pathways, and the mutations which cause CLAs result in hyperactive RAS signaling, either caused by impairing GTP hydrolysis or influencing nucleotide exchange by the RAS protein. So to investigate the effect hyperactive RAS signaling has on the development of lymphatic vessels, we had developed a mouse model and found that hyperactive RAS signaling in lymphatic endothelial cells during postnatal development stimulated lymphatic vessel invasion of bone and also impaired the formation of lymphatic valves, ultimately resulting in retrograde lymph flow and chylothorax. And by further characterizing this mouse model, we've been able to uncover what I think is some very interesting biology, where we have found that mutant KRAS has a greater effect on lymphatic vessels in newborn animals than it does in young adult mice. So in our mouse model, we used a PROX1-driven tamoxifen-inducible CRE to drive the expression of KRAS and mutant KRAS and green fluorescent protein in lymphatic endothelial cells. So after administration of tamoxifen to our mice, CRE recombinase excises the transcriptional stop sequences upstream of GFP and KRAS, resulting in the expression of these genes in lymphatic endothelial cells. So induced KRAS expression in newborn animals, we fed newborn mice tamoxifen and then collected ears from the mice 3 weeks later. And what we found was that the number of lymphatic branch points and number of lymphatic valves was significantly reduced in mutant animals. We also found that the diameter of lymphatic vessels was significantly increased in mutant animals compared to controls. In contrast, when we induced the expression of mutant KRAS in young adult animals and then collected ears either 3 or 6 weeks later, we found that there was no difference in the number of branch points or valves between control and mutant mice, and a modest increase in lymphatic vessel diameter. And I have some higher magnification images here to better illustrate this, where in newborn animals, you can see the control mice, nice appearing lymphatic vessels, and these intense regions of GFP staining, which represent the valves. And in our mutant animals, the absence of valves and enlarged lymphatic vessels when we express in newborns. But when we express in young adult mice, the lymphatic network appears relatively normal, where there are still valves in the mutant animals. So we think this suggests that there's this window where mutant KRAS has a greater effect on lymphatic vessels as the network is forming, rather than after the network has already formed. So to further investigate the effect of mutant KRAS on the development of lymphatic vessels, we performed a single-cell RNA-seq experiment. So in this experiment, we induced the expression of mutant KRAS and GFP in newborn animals, and then collected lungs from 21-day-old mice, digested these lungs to make a single-cell suspension, sorted the GFP-positive lymphatic endothelial cells, and then submitted these for single-cell RNA sequencing. And like what Hans went over, this is a UMAP, which is a way of displaying single-cell RNA-seq data. And I'm just going to walk through the data we have for our control animals first, before I go on to what we found with the mutants. So each individual dot represents an individual cell, and the cells are clustered together based on their transcriptional profile. So after quality control and filtering, we were able to get transcriptomes for approximately 1,300 control lymphatic endothelial cells. And these clustered into 5 distinct clusters. And I'm just showing on the right these violin plots for common lymphatic markers, showing that we have a pure population of lymphatic endothelium and an absence of blood endothelium in this population. We then set out to identify the subtype of lymphatic endothelial cells based on the expression of specific molecular markers. And after doing this, we identified 5 distinct clusters: the PTX3 positive capillary lymphatic endothelial cells, regular capillary endothelial cells, collecting endothelial cells, valve endothelial cells, and a population which expresses markers of both collecting endothelium and PTX3 endothelium. We then analyzed the transcriptional profile of these subtypes of lymphatic endothelial cells, and by doing so, we were able to identify additional unique molecular markers of the various LEC subtypes. And what I like about this heat map is that it further illustrates that this mixed population expresses both markers of collecting lymphatic endothelial cells as well as the PTX3 positive population. We then performed gene ontology analysis to identify GO terms enriched in the particular LEC subsets. And while we found many interesting genes, I just want to highlight what we found with the PTX3 population, where we found that genes associated with the regulation of GTPase activity, MAP kinase signaling, as well as VEGF signaling were enriched in this population. And we think that this could potentially raise the possibility that this PTX3 population could be sensitive to changes in RAS MAP kinase signaling. We then integrated our mutant and control data sets, and that's what I'm showing here with these this UMAP, where we have our control on the left and mutant data on the right. And through this integration, we were able to identify 6 subtypes of endothelial cells. So the 5 which I previously mentioned, the PTX3, the capillary, a mix, a collecting, and a valve, as well as now we have a proliferating population of lymphatic endothelial cells. And a few things jumped out to us right away. And the first thing was the reduction in the valve population in our mutant animals. And this is consistent with what we observed by immunofluorescence staining, where our KRAS mutant animals had a reduction in lymphatic valves. So we see that by immunofluorescence staining, and we also see this by single-cell RNA sequencing. The other thing that we noticed was that there appeared to be expansions of certain populations. So the PTX3 population, as well as the mixed population, which also all right. Okay, so like I was saying, the first thing that popped out to us was this reduction in the valve population. And then we see this expansion in 2 other populations. So to further characterize this, we measured the proportion of the different subtypes of lymphatic endothelial cells for both control and mutant animals. And in doing so, we were able to see that the PTX3 population was expanded, as well as the capillary mixed population was expanded in our mutant mice. And we have this reduction again in the valve population. And we also have a reduction in the collecting lymphatic endothelial cell population in these mice. And one thing that we also noted was that the collecting lymphatic endothelial cells in our mutant mice expressed markers of capillary endothelial cells and downregulated some of the collecting markers. So even though our mutant animals do have collecting LECs, they may be immature LECs and not have that full collecting lymphatic endothelial cell phenotype. And I think this is important given the finding that KRAS mutations can cause central conducting lymphatic anomaly, and we're seeing these defects in valves and collecting LECs with this KRAS mutation. We were also very intrigued by the expansion of the PTX3 population and how Hans very clearly described during his presentation. I'll just stick with this microphone for now. How he described in his presentation, activating mutations in PIK3CA result in the expansion of the PTX3 population, and this lymphangiocrin signaling influencing the microenvironment, and that targeting the microenvironment can impact the growth of lymphatic malformations. And we think a similar process could be happening for KRAS-induced lymphatic malformations, where during development, when a lymphangiogenic stimulus is present and you have a genetic mutation, that this results in hyperactivation of signaling in the lymphatic endothelial cells, which alters the cellular landscape of the vessel, in turn impacting lymphatic vessel function, resulting in lymph stasis, which can cause inflammation. Additionally, factors secreted by the lymphatic endothelial cells could promote this inflammation. And ultimately, the inflammation would provide additional lymphangiogenic stimuli to keep this cycle going. And we also think that any sort of infection or injury which would induce inflammation would provide these lymphangiogenic stimulus and maintain these malformations. So with respect to our future directions, we're working on validating our single-cell RNA sequencing data by immunofluorescent staining. We've recently acquired a PTX3 GFP transgenic mouse, which we're using to further characterize this relatively newly identified population of lymphatic endothelium. We've completed a single-cell RNA-seq experiment characterizing the immune cell populations in control and mutant animals. And we're continuing to investigate the molecular mechanisms regulating the loss of lymphatic valves in KRAS mutant mice. So to conclude, we've identified the cellular and transcriptional landscape of normal lung lymphatic endothelial cells, and we've demonstrated that hyperactive RAS signaling leads to the loss of valve lymphatic endothelial cells and the expansion of this PTX3 population. With that, I would like to thank the members of my lab who are all here at this meeting, especially Lorenzo Fernandez, who's been doing all of the bioinformatic analysis for the lab, our numerous collaborators, and our funding sources. And thank you for your attention, and I'm happy to answer your questions.